everyone. How are you doing? I hope everyone is doing good. So, welcome to another video of Molecular Basis of Inheritance. We are on chapter 4 of Maharashtra board and chapter 5 of the CBSE board. In this chapter, we were discussing about DNA replication and how DNA was discovered to be the genetic material. Okay, in the previous video we have covered the mechanism of DNA replication in detail. If you have not checked out the video, please check that out first. Now we are moving to the concept of protein synthesis, and we excuse me. We have understood a brief understanding about what is protein synthesis in the previous video. So now the protein synthesis that we discussed is made up of two parts, that is transcription and translation. So we will begin with transcription first. So in this video we are going to cover what is transcription and the processing of the mRNA that will be formed in after the transcription process. So the outcome of the transcription is the mRNA which is formed. Now this mRNA is known as messenger RNA, is known as heterogeneous nuclear RNA because it has certain sequences which are not required also, and that needs to be removed. So that is why this processing term is over here, that we will see in detail. You have a little bit of basis about transcription and translation, the whole protein synthesis from your lower standard. Okay, so any which ways we are going to do this in detail now. So let's begin. Transcription. So what is transcription? Basically, just to define in simple terms, is nothing but the copy of the genetic material. Okay, copying of the genetic material, that is the sequences or the information which is there on the DNA into RNA. Okay, so you have the double-stranded DNA. Yes, and let's say this particular segment is code. This is gene one. There is information regarding gene one. Let's say this gene one is making insulin hormone, for example. It is making insulin hormone. Okay, so this information to make insulin hormone is present on the DNA. To make the insulin hormone, for example, the machinery is there in the, this is in the nucleus. The machinery for making the hormone, or let's say let's take an enzyme over here, so that's more convenient. Um, okay, let's say an enzyme for glucose breakdown, which is hexokinase. Okay, this we have studied it in 11th standard. So this enzyme, this is the double-stranded DNA, and this region of the DNA is having the information. Let's say this is gene 32, for example. Okay, and this particular part of the DNA is responsible for making this enzyme. So the information is present in the nucleus, and the enzyme making machinery, that is a ribosome and other factors, are present in the cytoplasm. So it has to be copied over here from the nucleus and transported to the cytoplasm. So that work will be carried out by the mRNA, the RNA which will be formed. So RNA will be made over here, messenger RNA will be made over here according to whatever information is there. Okay, so it will copy the information and then it will transport outside the nucleus and take it to the cytoplasm where the protein synthesis will take place. Okay, so basically nothing but the copying of the genetic information from one strand of the DNA into the RNA. So you can see this blue double-stranded DNA. So the double-stranded DNA has to open up, just like in DNA replication. It will open up, and then so let's say this particular segment is the gene which is coding for a protein or an enzyme or some anything like that. So that particular strand will act as a stem tail strand of the DNA, and the transcription will take place. Okay, so an mRNA will be formed. Like you can see this pink one is the newly made mRNA. Okay, now because from DNA RNA is made, so we have studied in previous videos that there is no thymine in the RNA. So in case of RNA, in place of thymine there will be uracil. So we know that DNA 80 pairing is there. Adenine will always pair with thymine and guanine always pairs with by 3 hydrogen bonds with cytosine. But in RNA, so this is in case of DNA. Okay, but in RNA, adenine always pairs with same bond, 2 hydrogen bonds, but adenine will always pair with uracil. So in RNA, there is no thymine. In place of T, you have U, and the GC remains the same. Okay, G will pair with C with 3 hydrogen bonds. You can see over here. Okay, not here because here the information is getting copied. Yeah, here adenine. So the RNA is getting made. So here T will not be added. Which nucleotide will come? Uracil will come. So you can see over here U. Similarly, here A is there. So what complementary nucleotide will come? U will come. Okay, so these are the RNA nucleotides that will be brought in to make the mRNA. So adenine will always pair with uracil in the RNA. Okay, and the direction will always be in the 5' to 3' direction. Just like in DNA replication, the synthesis will begin from 5' end to 3' end, and similarly here also 5' end to 3' end will mark the beginning of the transcription. Okay, what is 5' end, what is 3' end? This we have discussed in the earlier first video of this chapter. If you have still not checked it out, please check it out. There I have explained in detail what is the 5' and 3' signifying. Okay, so the synthesis begins from 5' to 3'. So obviously the strand which is, see, let's say this is the 2 DNA strands. So this is 5 to 3 and this will be 3 to 5. Okay, so the synthesis has to begin from 5 to 3. Means what? The, let's say here the synthesis will begin. Okay, here. So 5 to 3 because the strands are made in opposite polarity. Okay, so this strand 3' to 5' will act as the template strand like this. Okay, so this strand will act as the template strand and this will be made in 5' to 3' direction. Okay, fine. And the 5' to 3' this strand will be the coding strand. Okay, so these terminologies you need to be aware of. This will be your template strand of the DNA. This will be the coding strand, and the mRNA will be made in 5' to 3' direction. Okay, can you see? This is the 5' and 1111 nucleotide is getting add up. So this is what you mean by from 5 it will start until 3, in the direction of 3 it will be made. This is in the direction of this strand is in the direction of 3' to 5'. Okay, and this is in the direction of 5' to 3'. Okay, so this is how the process of synthesis will begin. Now certain parts of the gene we need to understand. Okay, which will be termed as a transcription unit, as in which part of the DNA has to be transcribed. Okay, starting from where and ending where. So the start will always be known as the promoter. Like you have a train, okay, and the train always has an engine, right? The train has an engine, then you have the coaches, right? You have the coaches, and then you have the last coach. Okay, this is how a train will look like. So to pull the train, you have this engine in the beginning. Even sometimes you have engine at the end also to mark the beginning and the end of the uh, for the train to move. So this is, so the beginning will be the promoter and the end part will be the terminator. This will start the synthesis process. Promoter, start, the name itself is suggesting the function, and this will end. Okay, terminate means what? Stop, end. And this will be the part of the gene of the DNA that needs to be transcribed and made into RNA, mRNA. Fine. So the transcription, so this whole part we call it as a transcription unit. It is known as the transcription unit. 3 regions. Number 1, promoter, marks the beginning of the transcription, as we said. Okay, here the enzyme will bind. Obviously the making of the mRNA will require enzyme, just like replication of DNA requires DNA polymerase. So here polymerase enzyme will bind over here. Then after the enzyme binds over here, then this is the main part of the gene that will be actually transcribed, and finally you have the terminator which will stop the process of transcription. So the mRNA will be start making from here and it will stop here. So this is your mRNA. It will copy the information which is present here onto its strand. Single stranded mRNA will be formed. Okay, so this whole setup is known as transcription unit. Fine. Which enzyme will start the transcription process? So the enzyme RNA polymerase. Very important, you have to remember the name. Just like in DNA replication, what was the enzyme? DNA polymerase. So here, because there the DNA was being made out from DNA, daughter DNA strands are made. So DNA polymerase. Here from DNA, RNA polymerase is made. So RNA polymerase. Okay. Another name is DNA dependent RNA polymerase because the information is copied from the DNA into RNA. Okay, and the direction will always be, this enzyme will always start synthesizing into 5' to 3' direction. Okay, so the same thing, the polarity of 3' to 5' will act as a template strand. You have to remember that this will be your template strand. Okay, using this the mRNA will be made and this will be the coding strand. Okay, so 5' to 3' will act as a coding stand. We also call it as a misnomer since it does not code for anything. That is, we are using only one strand. The mRNA which will be made will have the same sequences of the coding strand only because the complementary pairing is there, right? So the coding strand has the sequence similar to RNA form, just what I just mentioned. Whatever coding strand has the sequences, the RNA which will be made after transcription will have the same information. Yes or no? For example, if you have, so we can go to the previous picture. This picture, if you see over here, okay, let's consider GGT. Okay, so GGT was pairing with CCA. So this was the DNA pairing. Now when the mRNA is made, G pairs with C, G pairs with C, and T pairs with A. So if you look at this sequence and if you look at the coding strand sequence, they are the same only. Na? This is nothing but the coding strand, which is also a misnomer, which will have the exact sequence of your mRNA which is being made. Okay, that is what we understand by coding strand has sequence similar to the RNA which is formed after transcription, except for change that in place of thymine only uracil will be there in the mRNA. That's it. No other difference. So this is the steps which are involved in the transcription process. We are going to see it a bit in detail. So beginning with the initiation. Okay, so three parts. Three steps will take place in the transcription process. Okay, just like in DNA replication, you had the starting origin of the DNA replication, then elongation wherein the daughter strand was elongated, and finally the stop. Okay, the end, the termination. So initiation, if you see over here, the template strand is here. This is the whole length of the DNA. Wherever the transcription will take place, the strand will open up. Okay, rewinding of the DNA will take place. Yes, rewinding of the DNA will take place. RNA polymerase will come and will bind to the promoter and it will start the unwinding of the DNA strand. Then the enzyme will recognize where the start site is, that is where is the initiation site, and the enzyme will synthesize the mRNA. Okay, and ahead of it, the unwinding of the DNA will be there. Okay, so this is the start site and this is the termination site. Then in the elongation part, what will happen? Just adding of the ribonucleotide. You can see over here the RNA nucleotides. that is ATP, adenosine triphosphate, uracil triphosphate, cytosine triphosphate. Just in the case we were discussing in DNA polymerase, one nucleotide comes and binds to the growing DNA's daughter strand. Similarly, one nucleotide will come and make this mRNA. Okay, so 5' to 3'. So this mRNA is getting made in this process. So this bubble will move forward. Yes, so the RNA polymerase, it will move. Okay, as and when it is transcribing, it will move along the DNA template from the strand from the 3' to 5' and produce the RNA transcript by adding nucleotides to the 3' end. Okay, so it will begin at the 5' and then in the direction of 3' it will start adding nucleotides, just like you see in this. This is the direction of the transcription, like you see in the representation. And when the enzyme encounters the stop site, okay, reaches the, when the RNA polymerase reaches the stop site, the RNA transcript is set free from the template. So the mRNA is ready now, and now it can freely now the mRNA is ready to be transported outside the site nucleus. You can see in this over here, in the nucleus transcription will happen and also processing will happen, and then the mRNA will transport, or it will be exported from the nucleus into the cytoplasm. This is the nuclear compartment. You can see the nuclear membrane over here. This is the nucleus and this is the cytoplasmic compartment. Then this mRNA will turn into, it will participate in the protein synthesis, and the information will be transferred in the form of protein. So these are the 3 steps that will take place in the transcription process. Okay, let's just go through it. So the initiation part, RNA polymerase will bind to the place where the transcription will start. Okay, there will be certain other factors also which will bind to help the RNA polymerase to start the transcription process. Okay, so something known as initiation factors like the sigma factors. Okay, this will help the RNA polymerase to recognize the start site of the transcription. Okay, then what will happen in elongation? RNA polymerase will use the nucleotides and it will take the nucleotides as the raw material. Okay, just like we were talking about the RNA DNA replication. Let's say you want to make a wall and this is one strand of the DNA and you require bricks and cement to make the wall, right? Similarly, one brick will act like a one nucleotide will resemble like one brick. So one nucleotide will be brought in. In case of DNA, you have all the nucleotides which will have adenine, thymine, guanine, and cytosine. And in case of mRNA, only the T nucleotide will be replaced by the uracil nucleotide. Okay, so polymerization will occur depending upon what is the sequence is there on the template strand. And finally, when the stop point is encountered by the enzyme, then there the transcription will stop. The other factors will come over there to help the RNA polymerase to stop the termination, to stop the transcription process. Okay, so these 3 steps will be involved in the process of transcription: initiation, elongation, and termination. Fine. So as the RNA polymerase proceeds to perform elongation, a short stretch of RNA remains bound on the enzyme. Then as the enzyme reaches the stop site, that is the termination region, this nascent RNA falls off. So whatever RNA has been made, it will get it detached from the setup, and then transcription will be stopped. Okay, just like this, you can see in the animation. See, the nucleotides are brought in, the twisting and the opening is brought about by other enzymes of the DNA, and this is how you can see this is happening in the nucleus. Transcription is happening in the nucleus. Okay, when the mRNA is getting made over here, the DNA will return to its position. So here is the termination. This is how the mRNA is here, and it will now move to the cytoplasm. Okay. Now, what happens to the mRNA that is made in this process? So now we have the mRNA. We have a specific name. We call it as hnRNA. What is hnRNA? genus nuclear RNA. Why don't we call it as the ready functional mRNA? Because at this point of stage, remember in the previous video we were talking about exons and introns. Okay, this mRNA, sorry, single stranded mRNA is made up of certain regions. Okay, certain sequences will be there which will be copied from the DNA. Okay, eukaryotic DNA has many such regions, many such sequences which do not code for any protein, which do not code for any amino acid. So they are known as introns. They are known as what? Introns. This we have discussed in detail in the previous video. If you have not checked it out, please go through that video to have better understanding. All the details have been discussed about introns and exons over there. Okay, so these non-coding regions has to be removed when the mRNA goes in the process of protein synthesis. So these not required sequences. Introns are basically not required sequences, and exons are the actual sequence which will code for the protein. So they have to undergo some post-transcriptional modification. We call it as post-transcriptional modification or processing of the hnRNA. So in eukaryotes, forms of RNA transcribed from the DNA are called primary transcripts. Okay, so now the mRNA which was made, we call it, what we will call it as primary transcript. Okay, this has to undergo certain changes, or you can say processing, or you can say maturation before becoming functional, before moving to the cytoplasm. Okay, as we see in this picture, yeah, as we see in this picture over here, the pre-mRNA, or you can just say over here, the primary transcript, okay, primary mRNA, or the mRNA which is there but non-functional, it will undergo processing in the nucleus only. There are few steps, 3 steps are there. Then mRNA will be made functional, and then it will migrate, it will be exported from the nucleus to the cytoplasm, okay, in the cell. So what are those steps? So you will see over here 3 steps involved in it. Okay, so number 1 is capping, number 2 is splicing, and number 3 is tailing. Very simple to understand. So you got your mRNA. You can see these orange and yellow lines, yellow and brownish lines. Okay, first step, first modification is adding of this cap. Okay, this cap is nothing but a nucleotide. The nucleotide is methyl guanosine triphosphate. Okay, methyl guanosine. Which nucleotide? Guanine or guanosine triphosphate. Okay, so this methyl guanosine triphosphate will be added at the 5' end. Okay, once the mRNA is ready, this capping will be added. Okay, basically this cap will act as a protection, will protect the mRNA from any attack, of its own enzymes. Okay, as the mRNA has to export, it has to travel from the nucleus to the cytoplasm. There might be enzymes of the cell itself which might attack this mRNA and try to, there might be some breaking or chopping off of these nucleotides. So to protect that, there will be a cap which will be attached, and there will also be a tail which will be attached. And this tail is made up of multiple adenine nucleotides. So many adenine nucleotides will be added at the 3' tail, a cap at the 5' and a tail. What is this tail? It is nothing but many adenine nucleotides which will be called as poly A tail. Poly means many. Okay, so now this is like you have this rope, and here also protection unit, and here also protection cap has been added. Okay, and then as we were talking about introns, which are not required, they do not code for anything. These are not required sequences, they do not code for any protein or any information, so they will be removed. Okay, So you can see spliceosome, they will just roll out and they will be pinched off. They will be pinched off, they will be cut, spliced, cut. Obviously, there will be certain enzyme machinery and factors which will come over here and they will cut and remove this. And now you are left only with the coding sequences which have the information to make the protein, which are your exons. Which are your exons. So you are now remaining only with your yellow part, that is exons. This is your functional mRNA. Now this is ready to be transported into the cytoplasm where the protein synthesis will take place. Clear? So capping, splicing and tailing. So that's all for this video. Read through the textbook. If you have any doubt, make a note of it and you can ask me. In the next video, we are going to cover about what is genetic code, uh, what are the features or characteristics of genetic code, and how mutations are, if the effect of mutations on the genetic code can affect the functioning or the metabolism of a cell. Okay, so thank you all. Take care. Bye bye. See you in the next video.